Hi guys, welcome to yet another lesson on the renal system here. This lesson we're going to cover renal handling of potassium, calcium, and phosphate. The first two videos will be potassium, how it's handled, then how it's regulated, and then calcium and phosphate in the last video. Let's get started. Let's start with potassium here. First of all, we take in quite a bit of potassium in our diet, and you'll notice that you do lose some in the feces, but the vast majority of the potassium that is lost from the body is through the urine here, and that's necessary to balance what you take in through the diet with what you get out of your body. So the vast majority of the exit of potassium is through the urine, and so this is reasonably important in terms of regulating that level. You will notice that although 98% of the potassium is actually stored in tissues, and of course a large proportion of your mass is skeletal muscle, and so that makes muscle a particularly important storage point. So potassium concentration very high in the cells, relatively low in the plasma. Nevertheless, it is the plasma concentration that is for the most part the most important thing that you need to worry about. So normal plasma concentration about 4 to 5. Hyperkalemia, high plasma potassium, low plasma potassium is known as hypokalemia. And it is important to regulate that potassium level, as you guys know, because you alter the resting membrane potential of cells when you alter the potassium equilibrium potential being by far the most important influence on what that membrane potential is. And so if you let it go either up or down, you really mess with the excitability of the cells, and you can cause serious problems. You should recognize that potassium is in fact shifted in and out of these cells, and that is reasonably important in terms of what that does to the extracellular potassium. So it isn't like it's shoved into the tissues and it's just sort of kept there. This plasma level is affected by how much of this potassium is getting out of the cells or being transported back in, and that is, as we will see, very relevant to pathology. So let's see here, we have a potassium load. So the plasma potassium bounces up because you've added potassium somehow to the body, either through the diet or through perhaps an IV infusion or something. And what happens immediately is you notice that almost all of that extra potassium is accumulated within the cells. So this is the percentage in the cells, or in the case of this yellow line, the percentage in the cells, and of course you accumulate the potassium in the cells pretty quickly, within a couple hours, okay? And that brings our plasma level back down where it belongs at about 4.5. That entrance and exit is regulated by two fairly important hormones, epinephrine and insulin. Both of these stimulate the sodium-potassium ATPase, which normally transports potassium into the cell, and so naturally enough you will decrease plasma potassium when you have epinephrine release or insulin. So in the case of epinephrine, if you get trauma, you'll get epinephrine release into the plasma, and that's fortunate because trauma often involves damage to the cells, and of course most of the potassium is in your cells, so you can get a reasonably, depending on the damage, you can get a reasonably substantial release of potassium into the plasma when you damage the cells. High epinephrine will help get that potassium back into the cells that aren't damaged, and get that plasma level back down toward normal in that case. In the case of insulin, of course, if you take in potassium after a meal, you need a way to get the potassium out of plasma, as I showed you in the last slide, get it into the cells. Insulin can help with that. In addition, you should recognize that high plasma potassium is a stimulus for insulin production. So it's sort of a self-correcting thing. High potassium in the plasma, more insulin brings potassium in the plasma back down as it's transported transported into the cells, okay? So it's sort of a auto-regulatory mechanism in that respect, okay? And you should recognize that when the plasma pH drops due to a higher than normal concentration of protons, you do get net potassium movement out of cells, so you will sometimes see a hyperkalemia along with a low pH. And that is something that we will worry about when we talk about acid-base. We'll worry about that more, but you should recognize here that there is a connection there between the two. And you should recognize, as I said earlier, you're not changing total body potassium when you simply shift potassium in and out of the cells. In order to do that, you need to either excrete it out of your feces, which is relatively minor, or you need to get it out in the urine. And so that's why regulation of excretion of potassium is reasonably important in the body. And that's why we're studying it here with the kidney, okay? So to achieve true balance, this diet has to be, whatever goes in has to go out, and the vast majority of what goes out through the urine. And you can see that happening here. So you remember initially with our potassium load, we got accumulation in the cells, and now you can see that that accumulated potassium is gradually excreted out through the urine, represented by this red line here, and the amount of that load that was stored in the cells gradually drops over a period of hours here. So this is ultimately the way that this influx is balanced by an outflux mostly through the urine, okay? All right, so the way that potassium is handled by the nephron, first of all, notice that it is freely filtered. So whatever's in the, whatever concentration of free potassium is in the plasma, you end up with the same concentration in Bowman's space, okay? 80% is reabsorbed in the proximal tubule, okay? We'll talk about that in a moment. And 10% is reabsorbed in the thick ascending loop of Henle, and that leaves 10% left in the distal nephron and the collecting duct. And this is where the control of excretion lies, actually primarily in this distal nephron, which is actually the distal convoluted tubule and the cortical collecting duct. And so this is where the excretion is regulated. You will note that in the medullary collecting duct, you always reabsorb, okay? So there's always potassium being reabsorbed into the medulla here. Where a lot of the major regulation takes place is in this distal nephron area, and that is where we'll concentrate. And notice that you can, depending upon the conditions, get excretion of potassium here or reabsorption. And you will see that in a moment, okay? And you will note that because of the variation in the handling here, you can get a huge variation of how much potassium is in the urine. In the case of a high plasma potassium, you can excrete as much as 150% of whatever was filtered in this Bowman's capsule. So this secretion can be very, very robust if it needs to be. On the other hand, if you've got low potassium, you can reabsorb to the point where you have practically no potassium left in the urine at all, okay? So, well, 2%, practically none, okay? So, going into more detail here, in the proximal tubule, again, 80% reabsorbed, so most of what's filtered reabsorbed there. This is paracellular through the tight junctions, which aren't so tight apparently, in that area. And it takes place because you're getting a lot of water reabsorption, as we learned earlier in the proximal tubule. That increases the tubular concentration of potassium. And when you increase the tubular concentration of potassium, it flows down its gradient from high concentration to low concentration paracellularly into the interstitium. So that's how most of this happens. It's just the fact that when you lose water, the concentration goes up. When the concentration goes up, potassium flows from high concentration to low concentration back into the interstitium, okay? And you will note that because the water reabsorption is dependent upon the sodium reabsorption in this area, water will follow the sodium. As we learned earlier, that this reabsorption of potassium is indirectly sodium dependent, okay? In the loop of Henle, as you guys know now, we have this sodium potassium to chloride transport going on. This is where a lot of our sodium reabsorption takes place. Beyond the proximal tubule. And so you lose about 10% of what's filtered here. That's reabsorbed in the loop of Henle, okay, through that transport. And that is also indirectly sodium dependent because this transporter doesn't work unless sodium is present. Its rate, the rate of transport here is actually sodium dependent as well, okay? All right. And finally, we have the handling in the distal nephron, and this is dependent upon the dietary potassium, or basically the plasma potassium. Okay, so in the case of low dietary potassium intake, you get reabsorption in this distal nephron, which is the distal convoluted tubule and the cortical collecting duct. Okay, and in that case, because you're getting reabsorption here through the intercalated cells in the cortical collecting duct. Remember, there are two types of cells, the principal cells and the intercalated cells. The intercalated cells in this area do the reabsorption. As you'll see in a moment, the principal cells do the secretion. So this is how this area can reabsorb under some conditions and secrete in others. It's because two different cell types are responsible. The intercalated cells for the reabsorption and the principal cells for the secretion in that area, okay? And so in this case, 2% here, 6% here, you add it all up, 98% of the dietary potassium is reabsorbed, okay? You get 2% left in the urine. On the other hand, if we have a normal or a high potassium diet, now we're getting secretion in this distal nephron. And this is primarily through the principal cells, which I'll show you in a moment, and the cortical collecting duct, okay? And again, always reabsorption in the medullary collecting duct. In this case, it is increased, it's about 20%. And that's because whatever you're secreting here, the reabsorption can't keep up, and you end up with a lot more potassium in the urine, okay? Okay, so how does this work? Well, because most of the secretion takes place in the principal cells in the cortical collecting duct, this is the only cellular mechanism we're going to consider here. It's the major one. The whole thing, as usual, is powered by the sodium-potassium pump, transporting sodium out and potassium in. You raise the cellular potassium, and then you have this apical potassium channel, which transports the potassium down this electrochemical gradient into the tubular lumen. So you're getting potassium secretion from the interstitium across the layer into the tubular lumen. You will note there is also a basolateral potassium channel. This does transport potassium back into the interstitium here, but ordinarily that potassium prefers to go through this apical channel because there is an electrochemical gradient across the entire epithelial layer from interstitial space to lumen, and that tends to make the potassium want to go across this apical membrane, and you get this secretion into the lumen, okay? When that channel's there, it gets secreted across, okay? All right, so we'll stop there, and we'll talk about control of potassium handling in the nephron in the next video. Thank you.